So this is the brief, the title of my topic, but I'll be going over different aspects. And I'll take this moment to acknowledge the funders, my research collaborators, the hardworking my lab group, especially BCRC, RR, and Saskatchewan area for their continuous support in their funding. My collaborators and my mentor, Dr. Surya Acharya, and my collaborators, Bill Bilugetu, Doug Tani, Dr. Stacy Singer, Dr. Gavin, Dr. Annie, and Dr. Youssef. They are my colleagues from the Eastern Canada, and Dr. Keshav Singh. And that's my lab picture, and where there are 2 of my graduate students and 3 technicians. And they are the ones who do the hard work, and I'm presenting the results. So there's a poster in the other room, and if you haven't, and if you are interested, it's in sainfoin grass mixture. It's a long-term study, so check it out. And if you have any questions, feel free to connect to me. And moving ahead, this is the brief outline of today's talk. I'll start a little bit by giving you the status of forage breeding in Canada, and then I'll be more specific. I'll talk about a little bit about my breeding program, and then I'll go more further specific. That's improving lipid content in forages. That can be a little bit of technical, but I hope you'll like it. So in this figure, I'm showing the on-farm yield of forage and other crops over the 60 years period. It was published in Bonneville, but what I'm trying to show here is the productivity of forages across this year. It's very low, and it's not only in Canada, it's everywhere in the world. We are about 3% gain per decade compared to like 15% in other crops like wheat and canola. And there's a serious decline over here after 1990s, and that could be because there's a conversion of the land and after effect of the green revolution that bring dwarf varieties. So the forages, they are converted to the marginal land. That could be the reason, but I'm trying to find out more data to complete all the way up to 2025. It's very hard to get all those data. And then moving ahead, this is the data from Austin et al., and it's a data in which cultivar. So the table is showing 1908 varieties over here and 1978 varieties. That's just after the Green Revolution. And what I'm trying to show here is what they are improving as they move from the earlier variety to the after Green Revolution varieties. They are just increasing their harvest index. Oops, sorry. So the harvest index increased from 35% to 50%, and there's not much room for them to improve. So if the wheat or any other cultivars that's going to improve, it's through, by increasing the total above biomass. So for forages, our economic product is both or all the above ground biomass. So when we are talking about the total photosynthesis on this one, in wheat, it's a green yield plus straw, but for forages, all the above ground biomass. And it's a very big challenge for us to improve the biomass, So yeah, and this is some kind of repetitions from Bill Gato's presentation in the morning. It takes a long time for a breeder to generate or to develop a cultivar. Generally speaking, it takes about 12 to 15 years. And the first section, the first part is the breeding, in which we develop populations, and then field trials, and then the commercialization takes about more than 4 years. So over a period of 12 to 15 years. And in the Lethbridge Breeding Program objective is to develop the climate-resilient cultivars suitable for Western Canadian region, and I will be focusing on conventional breeding to develop the conventional cultivars using the traditional method. But to improve the genetic gain, I will be using newer tools such as phenomics, and that's the use of the drones to capture the images and then do the prediction based on the phenomics. The other approach would be genomics, in which I would use genomic selection methods, GWAS method, QTL, and marker-assisted selection method. These methods are discussed briefly by Bill in the morning session, and the hope is it will reduce the breeding cycle and hence increase the genetic gain. So I'm focusing on these three crops. Alfalfa is it's grown everywhere in Canada, but sainfoin and cicer milkvetch, they are specific to the Western Canadian conditions. So current project includes sainfoin improvement project. We have long-term grass mixtures with alfalfa and sainfoin with grass mixtures. I have a poster over there. And I'm also looking at 220 different sainfoin accessions, learning about their diversity. The other one is cicer milkvetch, alfalfa compatibility study. And on that one, I'm using greenhouse stays to do the allelopathic effects, small plots to see the compatibility, and also the grazing experiment. The grazing experiment gonna start this summer. And finally, soot lipid improvement project, and this is in sainfoin and alfalfa, and I'll be talking about that. So before I move to the lipid improvement project, I'm showing here the list of all the alfalfa cultivars that are developed by public breeding programs in Canada. There's a list of 37. The yellow highlighted are the alfalfa that are developed by Lethbridge Breeding Program. And it's been since 2013. There's no any alfalfa cultivars from my program. But there are more than 500 cultivars registered in CFIA that mostly come from the breeding outside Canada. So here I want to introduce a new experimental alfalfa cultivar. It's named as LRC 4656, and it's a synthetic population developed by crossing 300 plants that were selected in 2016 from 20-acre block of AC Blue Jay alfalfa, and that was subjected to grazing for from 2009 to 2016. So since it's selected from the grazing, it's grazing tolerant, and it survived for multiple years. It's winter hardy, and it's high yielding. As you can see in this table, there's multiple locations and multiple years. And we're seeing almost 120% of the beaver at the different locations. The Lethbridge tests are mostly irrigated, and the Chain Lake, that's Alberta, that's not irrigated. And you can see the dry matter yield on this location is really low compared to what we have in Lethbridge. And what really excited me was the performance of this cultivar in Creston. So we seeded this one in 2019. This one is the yield data from a single cut. And then 2020, it was COVID year. We didn't harvest anything, or we didn't have any data on that one. In 2021, that's the second production year, we didn't see much difference. But after that, after second production year, this new experimental population that outperformed both aceblusae and beaver. In fact, the beaver is declining in its production after second production year, but the experimental alfalfa that I have, it's increasing over. So this one is really promising. Moving forward, I partitioned those biomass yield for different years, and then trying to see like how much each cut is contributing. So in 2023, and this is just a single cut, so in 2024, no, this is in 2021, the first cut, second cut, and third cut, they have kind of non-significant difference in the contribution. But as you move from 2022 onwards, second cut and third cut of the experimental line, it's higher than beaver. And that could be because this one has more regrowth potential, and that's contributing to the higher yield. Yeah, this is available for anyone interested, and I think it's in the website today. The deadline to express the interest is until March 2026. March 26, 2025. So moving on to the specific project, this one is the lipid improvement project, and a lot of work is done by my graduate student, Muhammad, who just recently graduated from University of Alberta. And the question, why we increase lipid in forages, and the answer is because there's a tax, right? More polluter, the polluter will pay more taxes. So ruminants, they contribute about 33% of the global anthropogenic methane emissions, and that reduces their productivity by 6 to 12%. And it is estimated that 1% increase in field lipid can reduce methane emissions by 5.6%. And for the favorite species, alfalfa, we are seeing only 1.67, and that's the lowest lipid content among all the forages species listed here. The method of evaluation on these may be different, but alfalfa, sainfoin, and these contents relatively lower compared to the other species. So how to improve the lipid content, or how to, one way is just to add supplement in the feed, but that's costly, impractical. The other one is improving the lipid in forages through plant breeding, and that would be more practical. So looking at the literature, there are several papers which has been doing work on improving the lipid content. And the conventional research that's going on in the US, it's in Illinois, and it's more than 100 years. And it's in grain as well. So they were able to increase the lipid content from 4.7% to 22% in 100 years. That's the conventional method. And there's transgenic approach as well, and in other crops like in ryegrass and the model alfalfa model crop, Medicago truncatula. So in this project, we tried to evaluate two different methods. The first method is just the conventional method using a chemical. We create a mutant, and then those mutations, they are random. They can occur everywhere in the genome, and that creates some high variability. The other approach, CRISPR-Cas, it's precise, and it has a limited and predictable variability. And the purpose of this research is to compare the two methods. And that's some debate concerns about the use of CRISPR-Cas, but this is for the research purpose, and we are making sure all these crosses and all these materials is grown in a controlled environment. So for the chemical treatment, we subject like 1,000 seeds of each species to the EMS, which is the chemical. And then after 14 days, they were transplanted to the bigger plot. And for the first set of evaluation, we used the lipid analysis through NIRS. That's the prediction-based method. And we also use the GCMS. This is the waste chemistry-based method. And then we conducted the RNA-6 experiment, and then crossing and developing the new populations. In the first round, so the EMS alfalfa and control alfalfa, we are seeing 7.7% increase in both alfalfa and sainfoin. So from this, we downsize it to 50 individuals, and then we make the selection both way. The high alfalfa, low alfalfa, high sainfoin, and low sainfoin. And that gives us like 20% increase in high lipid alfalfa and 7% decrease in low lipid alpha compared to the control, and 15%, almost 15% increase and decrease in high and low lipid sainfoin. And we further downsize it to 15 to 16 individuals over there, and we are seeing a significant 12% increase in high and 10 to 12% decrease in low for both sainfoin and alfalfa. So yeah, we break down into the fatty acid composition on this one, and all of the increase is on the beneficial fatty acids, such as oleic acid, linoleic acid, and linolenic acid. So we are seeing about 13% increase in the total beneficial fatty acids. Yeah, this is what compares to the canola seeds, and it's different. We have only around 1% something oleic acid in the soot lipid compared to 50 to 60% in canola seeds. It's just for the comparison. Moving forward, we want to know what is contributing to that, and then we did the RNA experiment for those who don't know what RNA sequencing is. And just a simple explanation of RNA sequencing here. There are two groups. We already have low groups and high group populations, and then we extracted the RNA. That's what produces the proteins, and then we sequence that one, and then just do the bioinformatics. So at the end, just to explain, suppose this is one gene, and if there is something happening between the two groups, the low one has higher rates, and then the high one has lower rates. That's just in a single gene. So we are looking for multiple genes and all across the genome. So on this one, we take two high lipid genotype, two low lipid genotype, and two controls for alfalfa. And then we just separate the stem and leaf tissues because they have different concentration of lipid content. and they were sequenced, finally mapped with the medic cargo Tranquitilla. Again, this is the model species and has more annotations of the genes. These are just the number of differentially expressed genes, and there are a lot. There's not a lot of sense to show all these, but and then we downsize. We look at deeper over there, and it's exciting to see these four genes. They are differentially expressed. That's biotin carboxyl carrier protein (BCCP), Cas3, cuticle2, and lipase-light protein. These genes has the functions listed here, and they are all contributing to the fatty acid synthesis. And just looking at how the gene count at the different level. Here I have 3 different levels, and then here's the gene count. And this is compared like with P less than 0.05, anything with asterisk, that's significant to the control. So I was expecting I would get something, the control between high and low, but it doesn't happen that way. But anyway, there's a lot of significant difference that warrants further validations of these genes to see if they are really involved in lipid metabolism. So the other methods is we are doing the same thing to improve through the gene editing methods. And we targeted 2 genes, PXA1 and STP1. This has demonstrated higher lipid in the model alfalfa plant. So, so moving on this one. So this is just the methods to develop the gene editing mutants. I'm not going to describe this in detail. And the results on this one. We got about 9 unique PXA edited and 11 unique STP edited genotype. The GEF, that's the genotype editing frequency. Because it's in autotetraploid, it has 4 alleles, and if anything it's something close to 50, that means 2 out of 4 alleles are edited. If it is 75, 3 out of 4, and if it is 25, then it's 1 out of 4. And this is the sequencing, and we are seeing like all deletions of the base pairs in the range of 3 to 6 base pair deletions. So now the results, are they really contributing to the change in the lipid synthesis or no? And this is for the PXA edited mutants. So this one is control, and there are a few that has significant difference, but the increase was only on this one, and in all other, that's a decrease. And on the stem tissue, we have 3 genotype that has a higher lipid. And then similar stories in STP1 mutant. So yeah, it's to interpret the results. We are not seeing like the consistent increase in lipid synthesis over here. And that could be because of different reasons, but I wanted to point it out here 2 different things. This one, as I mentioned earlier, is an autotetraploid, and we might have the mutation in only 1 allele, 2 allele, 3 allele, 4 allele, whatever. Like the gene might be fully functional if all alleles or all the homologous chromosomes are edited. The other one could be the frame-shift mutation over here. Just for an example, this is the original one. There's no edits. On this one, there's frame-shift mutation. That's the deletion of 1 nucleotide that leads to a different product. But when there is a non-frame shift deletion, just like if there's a deletion of 3 nucleotide, then we get the same product. That could be because of non-frame shift deletion as well. This is under investigations, and we are looking for that one. So next step, we want to cross those edits with 50% GEF to our elite cultivar, that's AC Blue Jay. And the progenies, we'll discard anything that has the genotype similar to AC Blue Jay, and we'll only take that has 50% edit. And then only on the second generation, we would be able to get this kind of genotype. That's 100% homozygote, and there we expect to have a higher lipid content. And moving forward, there's a lot of things to be done on this project. We'd like to enhance YUM2 EMS population up to YUM5 through conventional breeding. And then we'll also generate EMS half SIP families in YUM3 and evaluate genomic selection methods in multiple locations. We've identified the genes through RNA sequencing, and we want to validate that. And we'd also like to study the secondary metabolite change in our edited population. And finally, we'll also want to quantify the methane emissions, whether that's actually working or not. And for this, we have a 4-year funding project, and that's supporting a PhD student. That's starting in the fall 2025. And this is an advertisement as well. If there is anyone you know who is interested, you can pass on this information, give my contact. And with that, thank you everyone for listening, and if you have any questions, you can contact me, or just ask me over here. Thank you, thank you.
Thank you. Perfect. Does anybody have any questions for Harry while we're here?
So it's your question is what's the best variety to grow and graze? Yeah, I would say some of those cultivars that I listed there, AC Blue Jay, Longview, Grazeland. Yeah, so these cultivars, they are developed in Western Canada, and they have the persistence to grow on the long run, compared to the other cultivars, like the high-performance alfalfa cultivars. They are targeted to have higher performance just for a few years, and mostly they are targeting the hay.
Awesome.